Right. Okay, so good afternoon. My name is Kelly Racette, and I'm from the University of Florida. And today I'm going to be talking a little bit about some of my current dissertation research on hydrotropism and hydropatterning in peanut genotypes that exhibit transgenerational stress memory. So back in 1809, Jean-Baptiste Lamarck proposed two themes on the evolution of species. And the first was that organisms undergo adaptive changes in response to their environment. And the second was that those changes can then be inherited by subsequent generations. Now, historically, we've sort of abandoned those theories in favor of those of Darwin. But as we've already heard in this conference once, sometimes looking at old ideas with new eyes can lead to revelations. And that's sort of what's happened in plants recently, where we found that the parental exposure to an environmental stress can actually alter the phenotype of the next generation in the absence of DNA sequence changes. And so this process is known as transgenerational stress memory, and can occur through many different mechanisms, including epigenetics and maternal provisioning, though I won't be getting into those particular mechanisms in this talk. So we've actually observed the effects of transgenerational stress memory in peanut, where in a 2-year study of yield, we saw that the offspring of a water-stressed parent consistently produced more yield than the offspring of a fully irrigated parent in 5 different genotypes. And so after we had seen that particular pattern, we went looking for some other physiological traits that might be affected by this transgenerational stress memory. And given that we were looking at parents that had received water stress, one of the first things we wanted to look at was roots. And we saw in the first 12 days of growth under an optimal soil moisture environment, the offspring of these water-stressed parents differed in their root architecture from offspring of fully irrigated parents in 2 out of those same 5 genotypes. However, the architectural traits are not the only stress response that roots have to a heterogeneous soil-water environment. And so some other options for how plants might respond to stress in their root systems, in particular into water, are hydrotropism and hydropatterning, which act in a similar way to a well-documented phenomenon of nutrotropism and nutropatterning, where a plant encounters a high level of resource availability and either has directed growth towards that area or increases root proliferation in that area to better utilize that area of high resource availability. And as we just heard a very nice talk on the idea of linking this form and this function, linking root architecture to how roots actually function in taking up nutrients, I won't go into a lot of detail here, but I just want to emphasize the point that So I just want to emphasize a couple of points. The first being that a lot of our modeling approaches are frequently based on root level relationships and not on relationships of root architecture to whole plant systems or whole architectural systems. And the second is that these models do need to be validated by field level and non-destructed methods of quantifying root water uptake, especially because root function can vary over time based on the local environment that those roots are encountering in the soil. And so in this diagram just sort of represents 2 different options of a plant, and the question is sort of, can these 2 plants transpire at the same rate or uptake water at the same rate, even though they have different architectures in that area of high moisture content, because the plant on the right might be able to alter its function in order to maximize that water uptake. So that leads me to the specific objectives of this particular study, which were to evaluate the hydrotropism and hydropatterning effects in peanut genotypes that did have this transgenerational stress memory as a potential additional manifestation of that stress memory. And then the second was to relate those architectural traits to the water uptake relative to root length in a particular area of the soil, so a part of the soil that might have a higher water content, for example. So for this particular study, I used a seed source from a previous field trial that was conducted at the University of Florida, in which these 2 peanut genotypes that had a transgenerational stress memory effect were either exposed to a full irrigation or a water deficit. So that gave me 2 different types of progeny. Those that were originating from a non-stressed parent, or the ones that received full irrigation, and those that originated from the stressed parents, or the ones that had received that water deficit. We standardized all of these seeds by weight, so we wouldn't have any sort of effect of seed size on our root system during this early seedling stage. And we planted them in these rectangular tubes with a clear removable front plate that were all divided into 3 separate chambers, and each of those chambers was able to have a different water, a different amount of water in them. And an example of one of these tubes is shown here up on the upper right-hand corner. And so each one of those chambers either had 100 or 60% of the water holding capacity of the growing medium. And this gave us 4 different tube types. So basically we had 2 different germination environments. So these peanuts were either germinating in 100% of the water holding capacity or an optimal condition, or they were germinating in the 60% or water limited condition. And then the bottom layer of the tubes was either a homogeneous soil environment or divided into the 160%, giving the roots the opportunity to grow into or proliferate into the area of higher moisture or lower moisture. So these tubes, we grew at them 30 degrees from vertical in a greenhouse to encourage root growth along the clear front plate so that we could image these roots in situ over time. And we grew them for 21 days. And hydrotropism was determined using these pre-harvest images. And we determined if they were behaving hydrotropically by scoring whether the taproot had moved into the area of higher moisture or not, and by measuring the taproot growth angle on these images using image J. And you can see an example of one of these in situ images down here at the bottom right. And so that really allowed us to determine whether hydrotropic behavior was occurring or not. And then the Hydra Patterning we determined using WinRhizo analysis. So we harvested those roots and washed them in the standard way. And then we kept each chamber separate so that we could determine the total root length in each of those chambers on its own. And then for our second objective, we utilized the same tube system, but we took these tubes and put them into this very complicated looking experimental setup here. So we actually labeled one side of these divided chamber tubes with 5 atom% deuterium at planting. And then we attached this closed chamber here onto the top of the tube. And that was airtight, so that allowed us to collect the transpired water vapor in that chamber and run it directly through a Picaro, which gave us an amount of deuterium enrichment in the transpired air using cavity ring down spectroscopy. And so we also attached a infrared gas analyzer and a desiccant line to this closed chamber to keep the amount of water vapor in that chamber constant for each plant, to sort of standardize for differences in transpiration that might have been occurring at the given time of the measurement because of differences in leaf size or plant size or light availability or anything like that. And then we took that amount of deuterium enrichment and we standardized it for the root length in the tube to give us an idea of a relative root water uptake contribution of the roots in a particular section of the tube. So what did we find? Overall, we confirmed what we had seen in our previous studies. We had a significant impact of both genotype and parent stress history on our total root length in the entire tube. So we saw that one of our genotypes had more total root length overall, and that the offspring of the stressed parents had an increased total root length compared to the offspring of the non-stressed parents, just in that total root length of all 3 chambers. However, this difference in total root length was not associated with a difference in hydrotropic behaviors. As you can see here, in both of the germination environments, neither variety showed any response in the taproot to choice between the 100% or 60% bottom moisture. So there was no, whether the taproot moved into that higher moisture area or lower moisture area was completely random. So it did not appear to be behaving hydrotropically. Similarly, when we look at root angle, where a negative angle represents a movement away from the area of higher moisture and the positive angle represents a movement towards the area of higher moisture, we saw there was no difference in the average root angle of those taproots. So those taproots were definitely not sensing the moisture and going towards it or having any kind of hydrotropic behavior. Similarly, in our Hydra Patterning experiment, in both of our germination environments, neither variety or parent stress history had any impact on the relative root length between those two different chambers. So the high moisture and low moisture chambers had relatively the same amount of root length in there, so they had the same amount of patterning happening in them. However, although we didn't see any morphological changes in response to localized areas of higher moisture, this still we still have that question about the function of those particular roots. So as I said earlier, perhaps a area that has the same root length might be functioning differently. So in this case of this diagram, the black box and the red box have the same architecture, but we don't really know right now if they have the same function. And this is the very preliminary data from our stable isotopes experiment, which we are still in the process of analyzing. And you can see here that in our 60% germination environment, we did see a difference in the relative contribution of the root length in that higher moisture chamber between and the contribution of the root length in that lower moisture chamber, which is particularly interesting because that means that although there was no difference in the total root length between those two chambers, the roots in the higher moisture chamber are actually contributing more overall to the uptake of the entire plant. And then if you look over here at this 100% germination environment on the right, you can see that the roots in the bottom layer, so neither the high moisture chamber or the low moisture chamber was contributing very much to the uptake of the plant. So because that germination environment was not limiting, the plant was able to get most of its water from that top zone and didn't necessarily need to take up much water from the bottom zone. And then similarly, when we standardize that deuterium uptake by the total root length in the entire tube, we see the same pattern, but you can see that now the 100% germination environment becomes more apparent because this measurement is actually taking into account the root length of the entire tube, and that includes the root length in that top area. So once again, this is just emphasizing that in that 100% germination environment, we have a large contribution of the roots in that top zone and not a lot of contribution from the roots in the bottom zone. So this brings me to our conclusions. So we have no evidence of hydrotropic or hydropatterning behaviors in these two peanut genotypes. But the architectural differences and the differences that we saw in the architectures between the parent stress histories indicates that we probably need a little bit more screening. And there's been several other studies that have shown that the capacity for hydrotropic and hydropatterning behavior varies a lot between genotypes of the same species. So 2 genotypes might not be enough to really say if peanut has the capacity for hydrotropic or hydropatterning behavior. So we probably need to look at a lot more genotypes in order to answer that question. But we also found that this stable isotopes method can help us answer questions about root function relative to soil water content and what the architecture is in that particular section of the soil. And these preliminary results suggest that exposure to a water deficit could impact the relative contribution of these roots to plant water uptake. And so with that, I will open the floor for questions.
Any questions?